let me give you a sneak peek into the future. In the future, everything will be truly personalized. In the future, we will be designing one therapy for each individual patient. Personalization is now becoming a reality due to the advancements in artificial intelligence, or AI. AI is enabling us to develop cancer therapy that is specifically tailored to the individual patient based on the genetic code or the DNA of the individual. We can use the wealth of information contained within the DNA of the individual to tailor the therapy to that specific cancer patient. But now you may ask yourselves, how do we get from the DNA of an individual cancer patient to a personalized cancer therapy using AI? In order to understand this, let's take a step back and look at cancer therapy. In a traditional cancer therapy approach, such as chemotherapy, we're using a one-size-fits-all model. These types of therapies target cancer non-specifically, which causes side effects and is not always effective in fighting off the cancer. The challenge, however, is in cancer and in tumours, each tumour is unique, and we can argue that a more effective therapy can be developed if we treat the tumour as such. In a personalised approach, we want to target each patient uniquely. More importantly, we want to target each tumour uniquely. We basically want to look at the foreign elements that are displayed on the surface of the tumour cells and target these. These foreign elements arise from mutations that can be caused by, for example, environmental factors such as sun or smoking. Mutations are completely normal events in the human body, and we have developed many mechanisms to mitigate the potentially dangerous actions that these mutations can have. However, in cancer, these one or more mutational events have not been mitigated. And this can result in the development of a potentially malignant tumour. Tumour cells are disguising themselves as normal cells. This means that tumour cells are putting up a protective shield, allowing them to attract all the essential nutrients they need to grow, allowing them to ultimately grow out of control. The immune system cannot recognize the tumour cells as foreign. Our immune system consists of so-called T cells. And T cells constantly survey the body and monitor whether there's any danger lurking. They are experts in finding foreign intruders that are trying to invade the body, such as bacteria and virus such as the coronavirus, which I guess you are all experts in now in light of the pandemic. In cancer, the tumour should in principle be just as foreign to the human body as a virus is, but somehow it is not. The tumour goes unnoticed by the immune system. The tumour is in disguise. We want to design personalised cancer therapy that helps the immune system see the tumour. We want to help the immune system see the foreign elements on the surface of the tumour cells. And we want to boost an immune response that makes sure that we have the right T cells present and enough of the T cells present to actually fight off the tumour. In a personalised cancer therapy, what we want to do is we want to develop a therapy specifically designed based on the genetic code of the DNA of the individual patient. We want to bring these foreign elements that are on the surface of the tumour, we want to bring them out in the open. And the way we do that in a personalised cancer therapy approach is actually by providing the body with more of this foreign material, exactly as we would do in a vaccine against coronavirus. So, let me illustrate what these foreign elements actually are. These foreign elements are what we call neoepitopes, or new epitopes. And epitopes are small molecules that are displayed on the surface of our cells in complex with a specialized immune molecule called MHC. Neoepitopes, or new epitopes, have arisen from mutations in our DNA, and then they are displayed on the surface of the cells in complex with these MHC. Each cancer patient, or each tumour of each cancer patient, is defined by a unique set of neoepitopes. And also, each patient has a unique set of MHC. It's only a subset of the molecules that are bound to the MHC or displayed on the surface by MHC that are actually neoepitopes. And identifying these is a hugely complex task. Also, the immune system being able to see these is also a highly complex task for the immune system. Imagine that these neoepitopes are screaming at the top of their lungs, trying to inform the immune system that there is danger. The neoepitopes are trying to tell the immune system that there's something wrong with this cell, there's something wrong with this tumor cell, and that they should immediately kill the cell. The problem is, however, that this signal that the neoepitopes are trying to convey is drowning in other signals provided by the tumor cell, but also surrounding the tumor cell, that results in the neoepitope signal not being heard. What we want to do in a neoepitope-based personalized cancer therapy is we want to shift this balance and we want to amplify the danger signal, basically allowing the immune system to be able to hear the voices of the neoepitopes. We want to provide the neoepitopes with a megaphone in order for the immune system to be able to hear them better, in order to fight off the tumor. Let me try to visualize how we would design such a personalized cancer therapy. This you see here is a cancer patient. Or more specifically, this is the healthy DNA of a cancer patient. This is the DNA of the tumor of this specific cancer patient. What we want to do is we want to identify these mutations that are unique to the tumor of this specific patient. And the way in which we do that is overlay the DNA of the tumor with the DNA of the healthy tissue in order to isolate the mutations. This results in potentially a list of thousands of mutations. And what we want to do is we want to, based on these thousands of mutations, we want to identify which of those are actually displayed on the surface of cells and are able to interact with the immune system in order to inform that this tumor is actually a tumor cell. So what we want to do is we want to find the needle in the haystack. We want to find the neoepitopes that will be displayed on the surface. And this is a challenging task. In principle, what we want to do is we want to actually test each of these mutations in the lab. We want to test whether they are displayed on the cell surface, and we also want to test whether they have the ability to interact with the immune system. However, as you can imagine, doing this, testing all of these in the lab is a highly complex task, would take very long time, and is therefore not feasible. So we need other tools to identify these neoepitopes. And this is where AI comes into the picture. An AI algorithm in the context of neoepitope identification is trained on vast biological data sets that contain information about molecules that are displayed on the surface of cells, and we can use that information to identify our neoepitopes. An example of such data is that we take cells from lots and lots, thousands and thousands of different people. We then strip the surface of these cells for their epitopes. As you can imagine, this results in an extremely long list of epitopes. What we want to do with AI is we want to look at this really long list of epitopes, and we want the AI to identify patterns in these epitopes that will allow us to look at similar patterns in neoepitopes in order to figure out what is actually presented on the cell surface. What we do with our AI algorithm when we get a new patient and we want to see what neoepitopes are unique to this patient's tumor is that we use our AI, and the AI uses all its past experience that it's learned from all of the other data it's seen, and it uses that past experience to figure out for this specific patient which neoepitopes should we be bringing out into the open, which neoepitopes, which voices should we be amplifying in order for the immune system to be able to spot the tumor. Once we've identified our neoepitopes with our AI algorithm, we can go back into the lab and we can then make more of these neoepitopes synthetically. We can then put these neoepitopes into our personalized cancer therapy and give this back to the patient in order to amplify the danger signal and for the patient's own immune system to be able to fight off the cancer. Let me finish off by telling you a story about tomorrow based on many of the things that we can already do today. After many years in the sun, mostly without sun protection, my grandmother Harriet has developed skin cancer. She visits the hospital, and the hospital decides that a personalised cancer approach would be the way to go for her. A tumour biopsy and a blood sample is taken from my grandmother. And this is the material we need to design a personalised therapy specific for her. Once she is waiting for her therapy to be produced, we'll take a look behind the scenes and see how do we actually produce this personalised cancer therapy. We look at the DNA of the healthy tissue of my grandmother, and we look at the DNA of our tumour, and this is what we use to identify the computationally identify the mutational landscape of her tumour specifically. We can use the blood sample to identify the MHC, the immune molecules that are specific to her. Once we have this information, we can then use AI to extract the neoepitopes that are specific to her tumour. These computational steps can be achieved within a matter of hours. Once we've identified our neoepitopes, we can go back into the lab, synthesize them, make a personalised cancer therapy specific for my grandmother. And when she comes back to the hospital, she can be given her therapy. And after some time, my grandmother's own immune system will be able to fight off the cancer. Just imagine this world of tomorrow. A world where personalisation is the norm, and where each cancer patient can be treated specifically based on his or her tumour. A world where each cancer patient can be given a cancer treatment with no off-target toxicities and with high effectiveness in fighting off the foreignness in the body that is the cancer. Tailor-made, personalised cancer therapy driven by AI can potentially make this a reality. Thank you.
Hi, Emma. Thank you so much
Hello,
for that. Thank you very much for that very, very insightful and promising talk. First of all, I want to apologize. I didn't introduce you properly. This is Emma Jappe, researcher and PhD. I just wanted to correct that to make sure people know who you were. But listening to your talk makes me think, a lot of people here talk about cancer treatments a lot, and there's something just around the corner, and this is going to be solved very soon. And I guess I'm going to ask you the same question. Mm-hmm. How far are we with this kind of treatment? What's the bottleneck, if there is one? What's stopping us from going further with this?
Mm-hmm. So, as I said in the beginning, that this is the future. Personalisation is the future. And what I forgot to mention, actually, is that the future is much closer than you may think. And that this is actually where we are already. Wow. So we're already able to use AI specifically to look at DNA of individual patients. We're using AI in the field of medicine a lot. So we're both using it for diagnostics to see how should we treat a given patient, but now we're also starting to use it when we actually get a patient to actually design a therapy specifically for them. So we have two aspects of this. And this is already being tested. So this neoepitope-based therapy is already starting to be tested on humans. So we're actually much, much closer than potentially this presentation was
wow.
indicating.
That is really amazing. Thank you so much for your talk.